Okay. Again, folks, last week Dr. McAnalley spoke about his book, and Dr. Danny Sun also was there. Dr. Danny Sun is a plastic surgeon who is working with Dr. McAnalley now in this whole field that they're involved in. They're going to tell you some more. And Dr. Marvin, again, is on tonight to help officiate, direct the course of this discussion. And again, he's done quite a few webinars for us. Dr. Marvin, again, I just want to thank you for taking the time to be involved. And I'm going to hand it over to you.
And thank you, David. And it's a great, great pleasure you've given me to be involved in the seminar with Dr. McAnalley and Dr. Sun. I think most people know who Dr. McAnalley is, Bill McAnalley. He's the pioneer of the study of these wonderful monosaccharides, polysaccharides, and the oligosaccharides, and the physiology of which and how they interact in the body. So without me taking any more time from Dr. McAnalley, I want to give you the father of this whole technology from the beginning. It's my pleasure to introduce Dr. Bill McAnalley.
Thank you, Dr. Marvin. And we have two other authors here with me that I'm going to call on from time to time because we all wrote sections of this. And I think we're very fortunate to have Danny, who's not only a great plastic surgeon, he's a very good artist. And all the illustrations were done by Danny or Eric Boro, who's on my left side here. And what I'd like to emphasize is that the book is written for the layman. That is, we have an abstract and we have a summary, and then in the center we go through the science. A lot of people say, well, I told my doctor about this, or I told my brother who's a biochemist, and he said he wanted to see the science. So we have it summarized, and we go over it for the layperson. We have an abstract, and then at the end we have a summary. Then we have an introduction. Then we go through the references and the detail. You know, for example, we talk about the absorption and breakdown of ace mannan. So, and here's one of the diagrams that Danny helps to illustrate this. What happens is in our small intestine, we have pumps, little pumps that are sodium dependent, and they help pump mannose into the bloodstream. These have been identified, and they're able to find mannose and move it into the bloodstream. And why would nature design such a system if it wasn't important? And you know there's a lot more glucose in mannose. So with very, very low concentrations of mannose, the bacteria help digest and break down the polymannans into mannose here, and then they're pumped into the blood. And then, for example, here's some ace mannan, large polymannans. Bacteria release enzymes, break them down, and finally when you get a mannose, it's pumped across. And then we also know that pinocytosis can take some of the oligosaccharides and small polysaccharides directly into the bloodstream. The remainder, or most of the polysaccharides, are digested into small-chain fatty acids. And these are acetic acid, like vinegar, propionic acid, and butyric acid, and they feed the colon. So what you have is a system where fiber helps feed the colon, and this is a true symbiotic relationship. We present the studies that show if you take labeled ace mannan, that is, you grow it in a C13 atmosphere for carbon-13 instead of carbon-12, you can follow all the carbons in the ace mannan. And we were able to show, and we go through all the references and the studies on that, we were able to show 80% of the ace-mannan was metabolized as small-chain fatty acids, and 6.4% was metabolized as carbohydrates. 11.6% was absorbed and added to the mannose pool, and approximately 2% was lost in the urine, the cage washing, and the feces. But if you remember that the feces is 40 to 60% bacteria, the bacteria use the mannose to make their glycoproteins and glycolipids. So, very, very small percentage of the mannose is actually lost. That shows you how efficient, nature is. This kind of summarizes what happens. Ace-mannan is acted upon by probiotics, friendly bacteria. They go to, they break down into mannose oligosaccharides, and most of it's converted to small-chain fatty acids, around 80% of it. Now, these small-chain fatty acids are very important. This is just a summary of what the small-chain fatty acids do. We were talking about the importance of pH. They inhibit the growth of pH-sensitive organisms. They reduce the effect of alkaline toxic compounds. They reduce the loss of calcium and magnesium. They help you absorb calcium and magnesium. And it also increases the colon and hepatic portal vein flow. Then the 3-carbon propionic fatty acid, it helps relieve constipation, increases the same blood flow, and it will help prevent diarrhea. The butyrate increases blood flow, maintains the mucosal integrity. And what happens is the presence of these fatty acids cause your endothelial cells lining the colon to proliferate. So you have colon as well as the other areas of the small intestine increase. So nature's designed it where when you eat food, bacteria are digesting them. And during this digestion process, you may wear on the lining of the intestine. So the bacteria provide food to make new cells. And this also reduces the risk of malignancy and prevents diarrhea. So this, it shows some of the importance of butyrate. You notice it's involved in ulcerative colitis. What happens is patients with colitis, they have given them enemas of butyric acid, and they become asymptomatic. So what we have is a system where when you're first born, your mother gives you these beta-linked oligosaccharides in her milk. It's one of the main components. And then she gives you the friendly bacteria that make these small-chain fatty acids. And whenever you quit nursing, then you need to go to other sources like ace-mannan or other fibers to make sure that you get your needed probiotics. So here is a diagram showing ace-mannan. This follows the logical steps. And again, Danny did a great job. Bacteria release enzymes. They break down the large chain polymers into oligosaccharides, monosaccharides. They're pumped into the blood. And then cells like the liver, endothelial, other cells are able to pump these inside the cell, where in the endoplasmic reticulum, glycoproteins are made. And then in the liver, they're released back into the blood to help feed other cells. Glucose plus mannose can produce glucose, 6-phosphate, which is used in glycoproteins. And what's interesting is studies have shown that 75% of the mannose will come from your diet if you have it in your diet. Otherwise, you have to make it from glucose. So dietary mannose is used for this process despite the presence of 50 to 100 times higher glucose concentration in the blood. So why would nature design it that way if they didn't intend us to get those monosaccharides and polysaccharides? The other piece of evidence is if you take the polymannans, when they're pumped into the cells, they turn off the enzymes that convert glucose to mannose. And this enzyme is phosphomannose isomerase. So if you get your mannose from your blood, then the mannose itself inhibits the enzyme and blocks the phosphomannose isomerase, which we have down in the code. So we have a specific mannose transport on the cell surface and the ability to use that. Then it takes the mannose and inside the cells, all of the cells of our body use it to make glycoproteins and glycolipids for communication. Well, it's fascinating, how this dicol can flip in and out of the endoplasmic reticulum, and you start out, you have some sugars on the outside, some on the inside, and you make the glycoproteins that are the cellular words that are transferred to the cell surface and may be released or used within the cell to communicate to other cells. So this is a very, very wonderful system. And you talk about recombinant DNA, and then we talk about using bacteria to make proteins. Well, you can't use bacteria to make these glycoproteins because they're species specific. You can use bacteria to make a lot of your proteins, but not the glyco form. So the glyco is controlled with each species. And then, how is mannose moved? Well, we have mannose receptors, and mannose receptors are on macrophages, your liver and lymphatic endothelial cells, and your dendritic cells, and they are turned on by mannose. So the more mannose you have, the more of these receptors you make. And these receptors look for mannose, fucose, N-acetyl glucosamine, and they're calcium dependent. You can see that the sections 4, 5, and 7 has a very high affinity for mannose. So we have a mannose receptor, and here's a diagram illustrating the mannose receptors. So the mannose receptor, in this case, is in the cell membrane, and it uses calcium to help it bind to mannose, to little polymannans or glycoproteins with a terminal mannose, fucose, or N-acetyl glucosamine. So this is on the membrane of the macrophages, dendritic cells, a lot of endothelial cells, and when they bind, they form vesicles. They pinch off, they form these vesicles, and the pH starts to change, releases the mannose receptor, and the mannose receptor returns back to the surface where it can bind another one, and it goes through this cycle again and again. At any one time, around 10 to 30% of the mannose receptors are on the cell surface. The other 70 to 90% are inside. And what happens then, the mannose receptor returns, and the glycoprotein binds with a lysosome, which has enzymes that breaks this down into the individual components and amino acids, and this is used then to feed the cell and help make its own building blocks. The more mannose you have, the more receptors you have. And this also turns on nuclear factor Kb, which produces interleukin-1, tumor necrosis factor alpha, IL-12, 8, 6. These are cytokines that help in communicating. So this is, is one of the binding methods. Another is the liver produces proteins. Like kids, they first recognized this with children that were sick all the time. They found that they were missing this mannose-binding lectin or mannose-binding protein. It's an acute phase. In this case, it's a colectin, acute phase protein produced by the liver, and it binds bacteria, and it looks specifically for the mannose. So your liver produces this large protein that binds mannose, and it has a site here, a mannose-associated serine protease. And when this mass binds, it activates the production it turns on a cascade, which I'll show you here in just a minute. Then in this area of the mannose-binding protein, we have collagen, the collagen region, and then the actual carbohydrate recognition sites. And they also have a calcium binding site. So here is another picture of the collagen helix. But just think, it always amazes me, collagen on a pound-per-pound basis is stronger than steel. So inside your cell, you're holding these receptors with these collagen fibers. And the mannose-binding lectin or protein has a high affinity for mannose and fucose. And spacing is real important on the cells, whether it will bind or not. If these receptors are not spaced correctly, it won't bind. So cells can protect themselves, by having the normal, healthy arrangement. So here we've expanded it. I said if the serine proteinase, mannose-binding lectin-associated serine protease 2, binds to the mannose-binding proteins, it turns on the complement 4 and 5. It activates complement, and this complement will kill lots of bacteria and cancer cells. So this is nature's way of looking for defective cells or cells that should not be in your body. And then the complement, the fraction helps in the phagocytic process. Also, the more of these receptors you have, and by the way, guess how you get more mannose-binding proteins? You take more mannose. So if you take aloe, you get more mannose-binding proteins. You get more mannose-binding proteins, more of this happens. And the more you have, they turn on the actin in the cell skeleton. They rearrange it. The high abundance of this enables the macrophages to phagocytize bacteria, glycoproteins, and other mannose much faster. They also recruit other cytokines that bring in other immune cells. And just the presence of mannose-binding lectin, when it comes in contact with a cancer cell, it causes it to die. And it binds it because the cancer cell is sticking out these glycoproteins that a normal healthy cell shouldn't have. And then they die, and it helps the macrophages eat them. So if you look at this process, which we explain in detail, and we go through one of these in each chapter, the take-home message is that if you take aloe or other polysaccharides, you activate the system. And normally we eat McAnalley polysaccharides regularly. They say the hunter-gatherer used to eat a half pound to as much as 2 pounds of these fibers a day. We've processed that out of our modern diet because what we wanted is just the nutrients. We said we don't need all that fiber. But we used to get the fiber, and this was constantly turning the system on. So if you're eating a lot, it's telling your body, hey, I'm alive, and it turns on this audit mechanism. So let's look at macrophage stimulation by expanding. All I may do is just turn this over for a second to Eric, let him talk through the macrophage stimulation.
Well with asmanid, it goes through the same process, activating macrophages through the two mechanisms that Dr. Mackey just talked about, and that's with the mannose receptor and with the mannose binding lectin, both which can be found on or can affect the macrophage. So when you have asmanid come in, either as a polysaccharide form or when it's actually found on the surface of tumor cells, the mannose receptor and the mannose binding lectin are activated within the macrophage to either produce nitric oxide, tumor necrosis factor alpha, interleukin-1 beta, and interleukin-6. And that kind of creates an anti-tumor activity, anti-cancer activity. When the asmanid just comes in contact with macrophages themselves, you end up with the production of tumor necrosis factor alpha, interleukin-1 beta, and interleukin-6, which are essential, again, with natural leukemic activities within the body. And then you have the third alternative with altered viral glycoproteins, which end up actually exposing more of the mannose to macrophages and these other mechanisms of interaction. And you end up with production of interleukin-2 and interferon gamma, which again is an antiviral activity, and it ends up destroying the surface of these viruses and makes them less infective. So continuing with macrophage stimulation, macrophages themselves aren't just found within the body. They're created, they come from stem cells in the bone marrow. And once they're there, they're called monocytes. These monocytes actually travel through the blood and embed themselves within various tissues of the body. Where they end up determines what type of macrophage the egg will actually be, all these being referred to as resident macrophages. So if they end up in the liver, they're called Kupffer cells. If they're in the lungs, they become alveolar macrophages. If they're in the brain, they become microglia. And if they're just kind of found within tissues, they're dendritic cells.
You know what's fascinating about that, Eric, is if you take the macrophages, initially the monocyte can become any of these. Mm-hmm. And if you take one from the liver and inject it in the big toe, it'll get right back to the liver because it's already specialized. It knows it belongs in the liver. In the skin, they're called Langerhans cells. And then in the book, we had room for all the papers, but we couldn't hardly get all this on. But these are all articles showing asmanid stimulating macrophages in humans, rats, cats, dogs, mice, chickens. So asmanid, there's no question that it's a powerful stimulant of macrophages. And then this is a good illustration of how the macrophages work within themselves. There are actually three different actions. There's the antigen nonspecific mechanism, there's the classical mode action, and there's the alternative action. And what the take-home lesson here is the macrophage has all of these receptors built to recognize polymanins. And when they recognize them, they have a set course of reaction. Like the antigen nonspecific mechanism deals with these resident macrophages we just talked about, like the Kupffer cell. Whenever you turn them on with bacteria or ACE mannan or other sources of mannose, you produce CD14 and a toll receptor. They bind to that, and this expression turns on the nuclear factor Kb to produce free radicals like hydrogen peroxide, nitrous oxide, and interleukin 6A, and GM-CSF. That helps produce more macrophages. So what this is hey, we're in battle, so let's go and produce some more macrophages because we may need them. So the first time you see these foreign antigens, and in the case of ACE mannan, it doesn't do harm like bacteria. All it does is turn the immune system on. So if you can get fiber, that tells your body, hey, I've been eating. And since my food's probably not sterile, let's beef up our immune system in case my food has bacteria or other unwanted organisms. So this is a great, it's a beautiful design. This also causes expression of CD40 and CD80, which turns on T cells. And when these receptors are activated, they produce interferon gamma, and they go and help activate macrophages that have been recruited. Now, these macrophages that have been recruited and have been stimulated by interferon gamma, they're told, hey, we're in a serious war, so their reaction is much more drastic, much more severe. So when they come in contact with the bacteria or these other polysaccharides, they have an even stronger reaction. And they produce another major histocompatibility factor class 2, and this recruits T helper cells. Remember, in AIDS, it's the T helper cells that the virus kills. But this recruits T helper cells, and they in turn produce interferon gamma, and they turn on the cytotoxic D8s that help kill bacteria. And these T helper cells look for B cells that make antibodies, and they work together to select the right antibodies to attack and inactivate these bacteria. And then the T helper cell also produces interleukin-4, which can cause macrophages if you have a real big organism, like even some parasites. They come in and are attacked. They will cause these macrophages and or dendritic cells. They can cause them to fuse and become huge, giant cells capable of eating very large bad organisms. The IL-1 and R4 receptor is also activated, and so this causes the production of proline, polyamines, and collagen production. So this helps aid in wound healing. And that's kind of a quick run through. This illustrates macrophage activation. There's 3 different ways that it's activated, and mannose can work through all 3 of those ways and at the same time, depending on how much is there. Here's a another illustration talking about the interaction between hepatocytes and macrophages. Eric, would you like to walk us through this one? Or Danny?
Okay. In this particular slide, we're actually looking at the involvement of the mannose-binding and its role on hepatocytes and cell structure adhesion, defense, and immune activity of all cells. So we have hepatocytes that are actually producing the mannose-binding lectin, and once these receptors are actually binding to the mannose terminals of bacteria or large chunks of a mannose polysaccharide, it goes through the same process Dr. McAnalley talked about before, this cascade system where C4 and C2 end up binding along with the MASP2 and ends up causing lysis of abnormal cells, of bacteria, and at the same time bringing in other factors and other cells to be activated. One of these being the role of the mannose receptor itself. The mannose receptor, again, can be affected by other immunogenic factors like tumor necrosis factor alpha, interleukin-4, and of course the typical mannose terminals of bacteria and ACE and polysaccharides like ACE-mannan. Once the mannose receptor actually binds to the ACE-mannan molecule through a calcium-dependent bond, it's actually brought in, phenocetized, fused with a lysosome, and broken down and incorporated. So this just kind of shows that whole overview of the mannose receptor and the mannose-binding lectin working together to detect mannose residues and either destroy them if they're part of a cancerous cell or harmful microorganism like bacteria, or bring it in to be digested for the cell for its own use and the production of lipoproteins.
Okay, again, the take-home message from this is ACE-mannan are, like mushrooms and other polysaccharides that have been known to stimulate the immune system. Normally, when you eat these, you're going to get some microorganism with it, and this turns your whole immune system on and buzzes it up. So if you just take amber toast, you get your immune system really buzzed up. It becomes optimal. Then we'll look at macrophages and wound healing. And since Danny here is a plastic surgeon, I'm going to let him run through this path. And you'll notice that nature doesn't waste its tools. It uses the same cytokines, the same macrophages to help heal wounds. And we have 4 different stages that wound healing goes through. And Danny, why don't you walk us through this one?
Okay, a lot of different textbooks talk about the phase of wound healing, and most of them talk about 4 phases, and they are hemostasis, inflammation, proliferation, and remodeling. And if you can see this drawing, actually macrophages is involving all aspects of this wound healing process, and it's the only cell that is involving all aspects. And this is why macrophage is so important, and therefore come back to the understanding of why mannose is so important, because mannose stimulate macrophages to cause those things to happen. And therefore, with the understanding of this, and we understand why clinically we see when we use those important substance, it helps with the wound healing process. And as we can see from here, the fact that the bacteria has those mannose-rich receptors on the outside that stimulate the macrophages, and in turn, early on we see some of the picture that shows macrophages come back in and phagocytose those bacteria and take care of those problems. At the same time, the wound cause platelet, and then the platelet plug can cause a clotting to stabilize the wound, and that's the hemostasis process. Okay, and then macrophages come in to stimulate some of the cytokines and all that to cause the inflammation process, and then recruit more macrophages and come in and make this process even more acute to take care of this process, and then take care of all those organisms that needs to be taken care of. At the same time, stimulate other cascade mechanisms that we talked about before, and then ultimately come back with a complement system, again, to lyse some of those bacteria. And then from there, macrophages continue on its work to stimulate the wound healing by stimulating the collagen and then matrix formation, and therefore also fibroblast. All those things that comes in to cause the wound to generate angiogenesis, granulation tissue, and reepithelialization, and finally, forming a scar, and then later on go through the remodeling process.
Now, in chronic non-healing wounds, you have a metalloprotease, an enzyme that is in the last phase, and it's supposed to digest the glycocalyx that was laid down. And in chronic non-healing wounds, this is orchestrated by the macrophage. And they believe that the macrophage has lost its ability to control this, and that's why wounds don't heal. So it stands to reason that if you stimulate the macrophage, it can gain its control, and chronic non-healing wounds can heal. I know that when I first developed Carrington dermal wound gel a long time ago, we thought, let's prove that our theory is right, it works through the macrophages, and let's cause the non-healing wounds to heal. And we had people that had wounds for 7 and 8 years. The oldest one was a World War II veteran who had had a phosphorus grenade blow up in his face, and it had a wound in his face that hadn't healed for over 40 years. And that the mannins were able to activate the macrophages to cause that wound to heal. At that time, we didn't know that this protein existed. But what's wonderful is, as we learn more and more about wound healing, that just makes the picture more complete. And as Danny said, the macrophage is involved in all phases. In fact, there have been studies where they knock out all the different cells involved in wound healing, and you can knock out all the cells. That is, you can eliminate them, but they will still heal. But if you knock out the macrophage, the wounds won't heal. So the macrophage is the most important. If you can stimulate the macrophage, you should be able to stimulate wound healing. So this is one of our studies that we did at A&M several years ago. And we took normal, healthy rats, which people thought healed as fast as they could heal. These are young rats that were wounded. And here you have the untreated rats, and then these rats were given acemannan. And what was interesting, even day one, the wound, the contraction, the wound stayed contracted more with acemannan. So the average size of the wound was smaller than the control. Day 3, and these are just the average sizes or the area of the wounds. Day 6, and all of these are statistically significant. So, by day 10, the size of the wound is less than half. Day 8, it's less than half. And it's completely healed in day 13. The question then is, what about old rats? And what happened, I don't know if you remember the study where they were talking about fat rats lived a much shorter life, about 40% less than rats who had a controlled diet. So I thought, what are they going to do with those rats? Because to buy a 2 or 3 year old rat, they have to feed them 2 or 3 years. To get an old rat, they're very expensive. You spend hundreds or thousands of dollars just to get each one of the rats. So I called them and I asked them, what are you going to do with those rats? And sure enough, we were able to get them to donate them to our research. And here's 2-year-old rats who had a restricted diet. That is, rats are kind of unique. get big. As long as they have all the food they can eat, they get bigger and bigger until they die. If you see a little old rat, you know, you don't know for sure how old he is. But if you see a big rat, you know he's old. It took him a while to get that big. But you'll notice they heal much, much faster with Ace Mannan. And all of these are, the statistics is out of sight. 0.000. I think one of them is 0.000, but times 10 to the minus 12, you know, zeros. Then here is rats that got to eat all they wanted. It took a little longer, but even they healed much, much faster. So Ace Mannan took normal, healthy animals and caused them to heal faster, their wounds, and it was even more effective on fat old rats. So I guess if you're a fat old person, you ought to be sure to get some of this on your wound. Then Ace Mannan and viruses. We developed injectable Ace Mannan for Merrick's disease, which is a herpes-induced virus lymphoma. And we were able to make USDA's vaccine work. They had an antigen, but they had tried all of the myramyl dipeptide, the other immune stimulants, to try to get 88% protection or more. And with one injection, we were able to get 88% protection or more. So they were very, very happy with that. And I thought this would be great for kids because we could get kids protected a lot faster and, you know, get better coverage. But it's interesting, no one was interested because of the liability. No one would touch human vaccines. None of the companies wanted to take the liability because you have to control it against the current vaccination. And if the kid gets the disease, then they get sued for everything they have. And I didn't think about that, but no one was willing to take the risk. And then here is Ace Mannan's. These are studies, and in the textbook over here we have all the references showing its effect. Then Ace Mannan's also was approved injectably for cancer in dogs and cats. And it works the same way, you know, in any species. But, you know, I think it'd be nice. Eric, would you like to run through, say, part of this? Here we have all of our, first we have macrophages. We have, and these are like the Cooper cells, the Langerhans cells. These are dendritic cells. These are antigen-presenting cells. And a lot of them, they come from the macrophage line, but they specialize in presenting antigens and making antibodies, as we see here. And then we recruit macrophages, and they're turned on, as we mentioned earlier, they're turned on by interleukin, by gamma interferon and interleukin 2. So everything's involved. The liver is, if you have viruses, it produces the nanosbinding protein or ligand. So we have all four of the same cells. If you'll notice that the way nature designed our protection is these cells work together and they work in harmony. In this case, they're all concentrated on killing these cancer cells. And you can activate all of these by taking Ace Mannan, so they'll work better. And then for, you know, if you have that skeptic doctor or your friend who's a biochemist, Eric will run through that right quick for you.
Okay. With this mechanism, what we're going to be looking at is, again, the mannose receptor and the mannose binding lectin, and then the high levels of mannose residues on the terminal ends of the glycoproteins and glycosides of various cancer cells. With most of these cancer cells, what ends up happening is that you end up knocking off sialic acid off the terminal end, so it exposes mannose or fucose in greater quantities on these cells. So these are actually can be detected by the mannose receptors and the mannose binding lectins. These can serve to activate macrophages, and it ends up causing the release of various compounds that bring in more recruited macrophages like interleukin-2, interferon gamma. It also helps bring in dendritic cells, which help bring in helper T cells. All of these compounds work simultaneously at the same time to end up actually causing apoptosis of the cancer cells and getting rid of them, and actually being phagocytized by macrophages and other recruited macrophages. There's the production of various compounds with the interaction of mannose. So you have huge production of reactive oxygen species, nitric oxide, interleukin-8, interleukin-12, and other compounds that actually bring in macrophages. Large amounts of tumor necrosis factor alpha, interleukin-1, and interleukin-6 are also used to destroy infected cells and initiate phagocytosis of the destroyed cells. So as long as there's high levels of ace-mannan or any other polysaccharide, these cells are going to be activated and they're going to be on the search for any cells that express high levels of fucose, mannose, or N-acetylglucosamine, which are very rare within human cells in their terminal expression.
Okay, so normal healthy cells don't express the fucose, mannose, N-acetylglucosamine, but cancer cells do. And that's how the immune system finds them and destroys them. So, you know, all of these cells work in harmony. This is nature's way of destroying cancer cells. You know, you have to have tens of thousands of cancer cells before they can even be detected. It's been estimated that young children have, you know, around 10,000 new cancer cells, potentially atypical cancer cells every day, whereas adults have about 5,000. Children have more because they're growing much faster, and there's a random probability of these cells becoming aberrant. And so the faster you're growing, the more likely you are to have them. But this is nature's way of looking for them and killing them before you develop cancer. You know, if you have cancer, then you take chemotherapy, and that's after this is grown out of control. You know, what you need to do is get the right nutrition and help your body fight all these diseases naturally. So I think that covers the science, the summaries, and I'd like Dr. Marvin to comment. I know he read the abstracts and the summaries. And what do you think about a layman's ability to understand that? We went through the technical part. The lay explanation, I think, is very simple. What do you say, Doctor?
I agree with you 100%, Dr. McAnalley. I think this book is extremely well written and extremely easy for a layman to understand. The illustrations and the discussion and solution accompanying, explanation accompanying the illustrations make it very easy for a layman to read. And I think everybody that's even involved in any way with a healthcare provider status, whether they're physician or layman using nutrients to help people, they need to read this book. And it's an excellent book, easy to read, easy to understand for anybody, well illustrated, and it's a hardback, which means you'll have it around a long time. And I know some of it I've read 3 or 4 times just because it's so fascinating to me how all this works when any type of inhibitor comes into your body, whether it's a virus, a bacteria, fungus, or a cancer cell, and how our whole defense, just like we use defenses in the military, just the whole defense system works from 3 or 4 different angles to attack the offending intruder to your body that might cause you to have long-term severe problems. So I really think it's excellent, and we're going to have the questions now. Those that want to ask questions to start typing them in over on the chat log, and we'll ask Dr. McAnalley.
Could I know a lot of people have asked for us to get the book, so could we put that number up there? And
Yeah.
by the way, this time we have some people standing by to take orders. The phone was ringing off the wall last time, and we didn't think about people trying to order at night. So if we put the number up there, we do have people standing by to take orders.
Okay, I'll do that right away.
Yes,
While
we
you're
get
right-
this.
While you're writing your questions.
We get the number. People can call in and order that book tonight.
Can you see the screen?
Yeah.
There it is.
Great.
1-800-755-0044. And the books are $28.95 per book. But if you buy 10 books, you get them for $20 per book, and 18 books for $15 per book. I know there's a person I talked to today in Australia, and they've already ordered 18. And I know one in Arkansas that's already ordered 18 and is working on the second 18. So it's all before you, and this is an opportunity for anybody that uses any glyconutrient technology to help people, whether it's topical or internal, whatever it is. This is an essential. I was really impressed as a physician when I found out that macrophages had mannose receptors, because I didn't have a clue, because it's not taught in medical school. And this is why this book is so important, because your doctor, if he's open-minded, will be glad to read it. You can loan it to him to read, and it'll show him that this is for real. So, do we have any questions yet?
Yes, we do. Do you want me to just transfer them to your box, Dr.
That'll
Marvin?
be fine.
Okay.
Yes.
Yeah.
All right.
Dr. Marvin,
Yes.
just before the question, I think we sort of want to express an important concept. Our purpose is to make sure that people understand the miracle of body. Body has a intrinsic design that takes care of a lot of those things. Therefore, it is important for us to maintain those functions by giving them the right nutrient. And that's the only way to prevent disease and prevent cancer from happening. Of course, when disease or cancer has happened already, then we need to do some other intervention to control the disease process. But yet the most important thing, however, is to understand that the reason you have those disease or cancer is because your body is not functioning properly. And when that
Right.
happens, we need to find out what happened to it. What is the process that caused that? A lot of times it's due to nutrition. Therefore, we need to, correct that process so that the function come back. And then truly the miracle will start to happen again. And that's the purpose.
Okay.
I'm pretty sure you have gone into a lot of those study on the nutrigenomic in terms of how those nutrition, if it is properly instituted, then it really can take care of your body in a miraculous way.
Absolutely. When I was in the South African Institute of Medical Research, I found out that the Bantu doesn't have appendicitis. There's many colonic diseases they don't have. And I found out that at the time I was there, they were eating the corn, and they were eating the stalks. It's for sugar cane, grew along with the corn. And I used to do that at home. I'd take the piece of sugar cane stalk, and I'd peel off the hard surface, and then I'd chew the pulp, because it was very sweet, and then I'd spit it out. The difference is the Bantu swallow the pulp.
Yeah.
So they get about 100 grams or more of fiber a day, which substantiates what Dr. McAnalley had just been mentioning. Now, I have one question. It says, how do you know how much of the acemanin your body needs for any particular time, or does it matter whether it's for alopecia or whether it's for something else? Or in alopecia, should you rub it on your skin rather than take it in your body? That's one question.
Now you can do both, for any topical uses, but internally you ought to try to get a few grams every day. I like to take a heaping teaspoon twice a day. And then if you're looking at all the glyconutritionals, you really can't take too much.
No.
You can, usually I tell people to take, as much as they can comfortably afford. Because
Sure.
used to we would get several grams, or as much as half a pound or 2 pounds a day. I've had people, take 2 or 3 bottles of freeze-dried aloe, real stuff, and it doesn't hurt you. You can tell when you're taking too much because your billfold gets real thin.
Yes. Yes. It causes weight loss in the billfold.
That's right.
I have another question here. Is there ever any evidence showing that toxins interfere with the conversion of glucose to mannose? And I know that's a loaded question about the conversion of glucose to mannose.
Well, toxins work on all kinds of enzymes. I'm sure that there are several of them that affect that conversion.
Okay. I know that our body preferentially will take mannose in even if we have 80 to 100 times more glucose in the same vicinity. Is that right?
That's right.
Okay. If there's a question, if the book has all the data and diagrams as we are seeing in this webinar, and I can guarantee it does. It is every one of those in brilliant color and well explained are in the book. So
we start out with a lay explanation. If you don't want to know all of that science, then just read the summary.
Right.
But I wrote this book because I've lectured in this area hundreds of times, and a lot of people would like to have the book, when someone really questions them. That doesn't mean you need to know all this. I think if you just read the summaries, that's all you need, and most of that, they would amount to something like 20 pages, the
Right.
abstracts and the summaries, and you would be well informed. But if you want to get into the detail, I think this is the first time the detail has been put in such a clear and distinct way. And the way we built it is we started out talking about receptors, and then we talked about the cells that have receptors, and then how the cells use these receptors. So why would God make receptors for ace-mannan if he didn't intend on us using them?
That's right. And it's in breast milk.
Exactly.
And that proves it to me from the beginning. I believe this is an excellent book for a doctor who has an open mind to really for once see how this works. That's why I think it's so important. And I'll agree with you, the abstracts and the summaries are wonderful. In fact, I marked mine with pieces of colored paper for every summary, and I've read through those summaries many times and see how it would be for a layman to read this, and it's excellent. Easy to understand. Let's see. Is it safe for diabetics to use on a daily basis, and where do you get it? That's the question.
Well, there are several people that sell aloe. What you need to do is find one that that has analyzed theirs, and you know for sure that it has its ingredients. When you're dealing with liquids, it's hard to tell what's in there. They've had a reputation, whenever they're in short supply, they add a few more gallons of water. So, if you're dealing with the dry powder and it says on the label what's in there, then they're going to be in trouble with the FDA and the Federal Trade Commission if they don't have it in there. But I would ask, for the proof. I know Carrington, which I worked with, used to sell a real good product. And, I don't want to be a commercial for companies, but obviously there are several good sources. You could probably speak up if you know some good sources, Dr. Morgan.
Yes, I know some sources, and I tell you, if anybody wants to get a hold of me, I'll be glad to talk to them about those sources. I know it isn't maybe the best to talk to them on a webinar like this because that's pretty proprietary, and we might cause someone to go for one more than the other. But if anyone wants to get in contact with me, I'll be more than happy to share with you some of those. I have another question. I saw that hepatitis. Go ahead.
Yeah, this is an educational book, and for education, you can talk about anything.
Right.
You know, but when you start tying it to companies, that's where companies get in trouble.
Right. The proprietary end of it is you have to be professional is the bottom line. The question is, I saw that hepatitis C was listed. Does this mean that mannose is effective in killing that virus? Could you explain more about this? What else is necessary in eradicating this virus?
And, you know, one thing this does is, this does mean that these polymannose can help eradicate or destroy that virus. It's, if you can bind the virus and digest it, or keep it from being able to infect cells, it's hard for me to say you kill a virus because they're molecules, they're not alive. You know,
Right.
Bacteria are alive, you can kill bacteria, but you can't kill virus. But what you do is you inactivate them or you digest them and you make them harmless.
Right.
Certainly, those mechanisms were presented and it sure inactivates the virus.
You know, in medical school, I was told that a virus is a chemical that can reproduce by using another cell's reproductive mechanism.
That's what it is.
Yes.
And what's amazing is that these viruses, in order to infect the cell, they look for these glycoproteins. They look for a entrance point. And what happens in mother's milk with these oligosaccharides, they serve as binding sites. So the virus binds to these oligosaccharides, and then they can't bind and infect the cells.
Right.
So it
And
just binds, and they're just passed on through, you know, collected and digested by the lysozymes that we talked about.
Absolutely. And, you know, I think that's excellent, and it really explains in the book here how that works on viruses. Another question, is it safe? We never answered this. Is it safe for diabetics to use on a daily basis?
Yeah, you know, insulin, the reason we have diabetes is we get too much glucose. And only glucose and glucosamine use insulin. The other sugars have their own transport mechanism. So they don't need insulin. They don't cause insulin spikes.
Right.
Our culprit is glucose. So if you're dealing with a polymannose, it's not going to affect your diabetes at all.
No. The only way it could possibly affect it would be in a positive way.
Exactly.
Okay. Let me see. I got some more questions here. Did we cover the toxins interfering with the conversion? I think we covered that pretty well.
Yeah, basically toxins inactivate, you know, enzymes in general. And you know, we could look at a lot of toxins that would slow down these enzymes.
And if you want to talk about toxins, chemo is 100% toxins just about. And
Yeah.
I've seen people use these glyconutrients and glycoproteins when they're taking chemo and get much less side effects. Another question. I wasn't able to be a part of this discussion last week, and I would like to know if the book has all the data and diagrams. Yes, I can answer that for Dr. McAnalley. They're all there, and they're wonderfully done by Dr. Danny Sun.
Yeah, we just copied our diagrams out of the book we've been showing you.
Yes. One question. Did Dr. McAnalley discuss the difference of ingestion glycoproteins on an empty stomach versus consuming them with food during the webinar at any time?
No, but as a general rule, you know, when your body starts digesting food, it's receptive to food ingredients, and as a general rule, they recommend that you take dietary supplements with your food.
Because
Unlike drugs, a lot of times because food binds the drugs and blocks their absorption, they say you have to take them on an empty stomach. But that's not the case with foods. And there are pumps. In the intestine, it doesn't matter what else is in there, it's going to find that mannose and pump it into the bloodstream.
right. And what really impresses me, Dr. McAnalley, from the book, first time I've ever heard anybody mention this, and I've read many, many articles and books about glyconutrients, but the fact that mannose stimulates the availability of more mannose receptors.
Yes,
And
that is a powerful concept.
yes, it is. I think that was a pearl, so to speak, that I got out of the book.
Well, what we're doing, I'm back to making wound healing products. And so what you want to do is stimulate your ability to heal wounds before you have surgery.
Sure.
Or if you know that you're getting ready to fall off your bicycle and scratch yourself, you ought to take some extra polymannan, so that'll get your body ready for it.
Yes, and you mentioned that in the book where you had done a study where you put the topical AC mannan or polymannans on the skin before you introduced the bacteria, and the good effect that showed.
Yeah, that's probably why, it's amazing how you look at Alexander the Great. Aloe's been known and talked about as a miracle plant for thousands of years.
Right.
And they talk about Alexander the Great. Anytime someone had a cut, he had fresh aloe plants that they used to take a leaf, cut it off, and wrap it and tie it to the wound. And I can understand now how that, would bind infective organisms. It would turn on all the macrophages. It would turn on the immune system optimally. And that's why it's been used as a healing plant for, thousands of years. The only thing is a lot of products have all the ace mannan taken out of them.
Yes.
I know one company's Aloe, and I mentioned that in the first chapter, and he said it tastes like spring water. And when we analyze it, it is spring water.
Right.
All the aloe components are gone.
I also heard the reason why Alexander the Great conquered the world at that time was due to wagons that he took with his troops full of pots of aloe. And also I read I don't know, may have been in this book, the reason why he went to Egypt was to get aloe from a small island there.
Mm-hmm.
Yeah, the island of Socotra, and we talked about that in the first
Yes.
chapter.
Right. And I was really excited.
And proof of that is he used to, when he would conquer one country, he would take people from the previous country and move them over to help rule that country.
Right.
And so what he did, whenever he conquered the island of Socotra, he moved some Greeks that spoke a particular form of Greek, and they're still there in that same area today. And they still speak that dialect of Greek, the Ionic Greeks.
Terrific. I have another question, and I know the person that asked this. How long should I expect aloe to restore brain cells that were damaged in a massive stroke?
So what we've learned now is that we're constantly producing new, I was told you get all the brain cells
Yes.
you have, you're not going to need more. We now know that's not true. And we showed you the pathway where taking aloe is going to constantly produce new stem cells. I think it'll produce them your whole life and gradually put them back in the right place.
I remembered eating dinner one night with Alex Amolichuk, who had destroyed 40% of his brain in a cerebral aneurysm bleed. And most people on this line, I'm sure, has read about him and heard about him, but I visited with him, and in a matter of about a year and 7 months, he had replaced that 40% he'd lost and was back teaching in a medical school in Toronto. So that's pretty good proof of that, I'd say.
Mm-hmm.
Now, okay, some people want to know how much to take, and we've pretty well covered that because Dr. McAnalley said he takes a heaping teaspoon twice a day of the acemanin.
Yeah. I think some of the point that we sort of want to talk about is that, you want to look at your body as a natural process. That
Sure.
means that, you, like last time Dr. McAnalley has talked about, you don't take, 10 breaths of air and then stop breathing for the rest of the time. So, this is why we come up with, the process of you should eat multiple meals, small meals, all the time. And due to the fact that our lifestyle now today, because of the work and all the schedule, it is impossible to do that. That's why we have 3 meals, or sometimes even 2 meals, or some people don't have time, only take 1 meal a day. And those are not necessarily the natural process. The old time, we're hunter-gatherers. We walk around and we pick up whatever food we have, and we continue to eat all the time. And that's how body gets supplied all the time. And interestingly enough, that most of the food, the way we process them by chewing them, and the body processes them in a natural way, instead of processing them first, then we actually get a full nutrient in a time-release fashion. And therefore, your body continues to get those nutrients all the time. Therefore, we replenish your body all the time. So, if people wanted to take those nutrients, if they can, then they should take those nutrients throughout the day, as much as they can. Of course, you don't want to stress yourself out. So, however you can fit into your schedule, and this is how you should do them.
Great. Here's a question I have for Dr. McAnalley or Dr. Sun. How can people digest freshly picked aloe from home gardens? How much can they safely digest?
Well, the thing is, that they know how to process it. Commercially, and on these islands, these leaves grow to weigh 2 to 3 pounds. And if you take a 2-pound leaf, and that's most companies have a minimum of a 2-pound leaf. That's a big leaf. It's like 2 feet long, and you can peel off the cheese cutter. You can peel off the green rind, and then wash off the gel. And that gel is 0.2% acemanin. So if you have, a pound of it, you're going to get 0.2 of a pound. 0.2% of a pound of acemanin, which if you eat a pound every day, that would probably be good for you. I think you'd be doing well.
Okay. I don't know exactly how it tastes, but that would be one of the problems. The little stickers on that leaf would also bother me. Peel enough. So.
Yeah, but it can certainly be done, and it needs to be done all the time.
Okay, here's a question. I know there's been 2 double-blind studies on. I'll give it to Dr. McAnalley. Does any other supplement aid in transport of acemanin glyconutrients past the blood-brain barrier and into the brain to help increase brain function?
Could you just a second? I was just thinking of another analogy while you were asking that question. But if you take about a quart, which a little more than a quart, which is a liter, that weighs 1,000 grams, and of that 1,000 grams, which is more than a quart, you get about 2 grams of ace-mannan. So 2 grams of ace-mannan, would fit in a teaspoon.
Right.
So you don't have to take a whole lot, but when you're talking about ace-mannan, you have to take roughly 2,000 pounds of gel of the leaves to get 2 pounds of ace-mannan.
Right.
But actually, one of the problems of we processing them ourselves is the fact that if we do not process them properly, the enzyme is going to digest the ace-mannan. So therefore, you really have to eat it fresh, and if you keep it, then some of those ace-mannan will be gone by the time you eat them. So those are the problematic things for us to do it ourselves.
Right. And that enzyme is there from the time you cut off the leaf. It starts to activate to destroy the ace-mannan, right?
Right.
Okay. Here's the question again. Does any other supplement aid in transport of ace-mannan glyconutrients past the blood-brain barrier and into the brain to help increase brain function?
I'm really not aware of any.
Well, I know the study in the University of Arkansas where they took 120 freshman students in the college there and did a double-blind placebo-controlled study, and on all parameters of cognition, they were increased. And that was done by a husband and wife research team. And I read just recently where there was another study done for mental alertness and all types of different facets of cognition that were increased in that group of students given these glyconutrients. So
Yes, I agree. There's no question that in fact, on page 69 I show how much of the label, got into the brain. So there's no question that ace-mannan gets into the brain taken orally. You get, you get quite a bit of it in the brain. And there's no question that it enhances their IQ. So, but there have been, 2 very good studies that demonstrate that.
Yeah. But as far as which nutrient or enzyme, we don't know any study have shown anything that aids that process.
Yeah.
Right.
The scientific question is, what can we do to make it better? And if I knew it, I'd be doing it.
That's right. And, it works because the body needs it. And Dr. McAnalley covered in the first webinar the fact that you can tell from babies breastfed 3 months, 6 months, and 9 months, you can tell a difference in IQ between the ones fed for 9 months from the breast and ones fed for 6 months from the breast. So we know that it does get into the brain.
You can tell the difference in IQ, if they're 18 years old. So,
Right.
So it definitely makes a difference.
Absolutely. Okay, let me see if I got any more questions here. Oh, the question was, why aren't foods enriched with mannans? And I would include baby foods there. So that's for either one, Dr. McAnalley or Dr. Sun.
Well, Dr. Sun, I've been thinking about making the product that way. It's amazing how something's known, and as well as we know this, that would be a great gift, I think, to mankind.
Sure.
If anyone would like to come up with a few $1 million, we'd love to start making that product.
Okay. Sounds great. Can mannose help in skin cancers, topically or internally? Nobody can tell me if the skin blocks are effective. I don't understand that skin block thing.
Yeah,
but
me
they want to
neither.
know.
Maybe what they're asking is that sunblock, whether that truly is effective or not by preventing skin cancer. I think that's what they were talking about.
Okay.
Well, ace-mannan is not a sunblock.
Right.
So you know, the best thing is to stay covered up, like farmers. The old farmers learned a long time ago in the summertime, or when you're out flying, wear long sleeves and gloves.
Absolutely. My granddad used to wear those rubber bands around his long shirt sleeves to keep the sun off of him, and he wore them every day all summer long. It can help in skin cancers then, both topically or orally. Is that right?
The question is that it certainly can help because what happened to the skin when it becomes abnormal is because of the external factor. So of course, the first thing is we need to remove the external factor that is the cause of the problem, and then we use it to enhance the body function for recovery, and in that way, yes, it will help.
Uh-huh. Okay. I have another question here, and I know one of you can answer this real easy. Can we have the book shipped to Australia? Is there an agent in Australia, or how do we order them? Do we use this phone number?
Yes. Go ahead, Dan.
Actually, we have done some research on that subject, and I need to confirm that. I just talked to the United States Postal Service. They do have an M-bag shipping process, which is probably by far the cheapest for books. And then if those people in Australia can gather together and buying bulk 18 at a time, then it will ship a lot easier with protection. Then when they get there, it will probably be more intact because what they do is they put it in a bag and then they ship it. So if we can package it in a box and well protected, then it will be a lot better. For 18 books. For 18 books right now, I think the shipping handling is about $60. So that's so far the cheapest way we can find to ship them to Australia.
Okay. The number again is 1-800-755-0044. And the price of the book is $28.97 if you order one book. If you order 10, they're $20 per book. If you order 18, that's why we've been mentioning 18, you get them for $15 per book. And as Danny said, that's the best way to ship them to Australia.
So if you're in Australia and you get together and you buy 18 books, the shipping right now looks like it's going to add $4 more to it, so they would be $19.
Okay, $19 including shipping.
Right.
Okay. Let's see if we got some more questions here. It says here, how does the acetaminophen affect one with autoimmune disease?
Well, the reason you have an autoimmune disease is your cells aren't communicating properly. And if you'll go through those diagrams, that we went through, we talk about that in our chapters in there. And the way it works is it just restores normal communication.
And by communication, Dr. McAnalley means the expression of the terminal residues. So a lot of what's going on, especially with inflammatory autoimmune diseases, is that you end up expressing lots of the mannose or fucose residues at the terminal ends of your glycoproteins. These are recognized by macrophages, and they release all the different compounds we just talked about. So it thinks they're cancerous cells or thinks that there are foreign organisms or compounds that aren't supposed to be there.
So the thing to do is get your cells communicating properly.
Take your mannose.
Yeah.
Okay. Now the next question, does mannose stimulate stem cells, and how many can be stimulated? We've kind of answered that, but we never got down to the how many.
Well, only God knows how many.
Right.
But, if you look at your friendly bacteria, and if you read through the chapter we go through and show how, the average person has 10 friendly bacteria working for every cell in his body. and you have a lot of cells. That means you have a lot more bacteria working and helping. But everything has feedback and feed forward. So by taking the right nutrients, if you need a lot more stem cells, it'll make them. And then when you have enough, it feeds back and says, that's enough. I don't need any more. So the great thing about using foods is they're under control. They don't overproduce, and they will help you produce the right amount.
Excellent. Excellent.
Yeah. And I think the body is a dynamic process, and it involving a lot of different aspects. Certainly when you're healthy, everything is functioning properly. And when something goes wrong, the body, its innate function is trying to convert you back to normal, and that's what it's trying to do. But certainly there's also limitation to what it can do if your body has gotten to a point that it really doesn't have any capacity of doing anything. Then it's really too late. This is why we always tell people to start this process as early as possible to make sure that your body is always healthy all the time.
And
Okay.
back to the book, I buy every book on aloe that comes to market. And hardback books like ours, others that don't have anywhere near the diagrams and everything, cost $100 or thereabouts. And what we wanted to do is make these affordable to everyone and make them where, they could even be given as good gifts. But when you look at this is something you wouldn't mind having, on your coffee table or giving to a friend.
Sure. It's very well done, and I'll agree 100%. I've seen books that have illustrations in them that aren't colored or diagrammed as simplistic as these are and well explained, sell for $150. I have a grandson in medical school, and his books run way up there that aren't even illustrated as well as this one. Here's a question. Can we quote the stem cell study where it showed aloe increases stem cell production?
You can quote any study you want to. That's why they're in that book. And these are, peer-reviewed studies. And if you'll notice, we may list 30 of them on a page, and we could have easily listed 300.
Absolutely.
Normally, all you need is one or two good studies, and this should be overpowering. I know I would hear people say, well, there's no peer-reviewed studies to support this. Well, here they all are.
I've heard that many times, Dr. McAnalley. A question here. What do you mean by cancer cells express fucose and a couple other saccharides? What does the word express mean?
Okay, normally, when a cell, goes awry or becomes atypical, like, let's take prostate or breast cancer, whenever a cell becomes abnormal or atypical and is on its way to becoming cancerous, it will start to express glycoproteins that tell the immune system that I am out of control. And the immune system looks at it just like we showed you there and kills it. So you never have cancer because it kills it before it ever starts. And those are atypical cells that express that is on the outer part of the glycoprotein. There's fucose, mannose, or glucosamine. And you can do a search on cancer antigens, and you'll see they have fucose. That's real common in breast cancer and GI cancer. They express fucose on the end. And normally a healthy cell does not do that. It covers it up with sialic acid or one of the other sugars, so it can't be seen. So when a cell forgets what it's supposed to be doing, and I think that nature actually just fixed it that way. If it starts to go out of control, it's recognized and healed before you get sick. Because like I said, when you get cancer, you've already got a lot of it. You want to control this naturally when these cells aren't cancer, when they're just atypical.
That's right.
And
Well, go ahead.
yeah, you recognize them by expressing these sugars. By expressing them, they mean on the end of the cell. Like if you look at my diagram, you'll see the glycoproteins coming out from the cell like little hairs. And if they have a fucose on the end or a mannose on the very end of it, instead of inside on the start of it, then it's an abnormal cell.
Great. Okay, another question. Are there studies showing ingested mannose make it to a cell surface glycoprotein?
Well, the study, there's not a lot of studies, like that, because to show that happens, we have to give you radioactive mannose. And I don't want my kids taking radioactive mannose. I don't want to take radioactive mannose. But we did present studies where it was given to rats and dogs. And it shows that, and that's on our around page 60. We go through those two studies where they gave labeled sugar, and it ends up virtually in all of the cells in the body.
Absolutely.
And we even have the amounts that get there, the percentages.
It's very well explained in the book, and that it emphasizes the fact that I believe everybody that has anything in the way of trying to help people with this technology needs to buy this book. And, it just answers so many questions, and it did for me, and I've read all kinds of books, but this is all congested into one book. And you can carry it around with you easily in a briefcase and refer to it. Now, a question here. Does soy lecithin help assist in the ingestion of ace-mannan in any way?
I've heard that by several people, but I haven't seen any papers that show that.
The only
I
person...
think probably if they're looking at how they feel, and I think soy makes people feel better too. I've heard people say if you'll take ace-mannan with soy or ace-mannan with this, it works better. Well, I don't know that the ace-mannan works any better, but probably the other things you took with it also help. So you feel better.
Sure. Absolutely. I really promote that in people that are very sick. How much of the glyconutrients, more I assume, should a smoker be taking that wants to take ace-mannan?
I think if you're going to smoke, you ought to take more. Again, I take, a heaping teaspoon. That's because I don't have tablespoons. But I try to take a tablespoon, if I had a real measuring spoon, at least twice a day. And Danny and I will take a lot. We probably take an ounce or 2 ounces,
Yeah, actually, Bill and I take a lot of those ace-mannan, in the form of powder, and we go through those tubs,
1 a week.
well, yeah, at least 1 a week.
Sounds great. People that smoke just should take more.
Now they need more because they got more damage.
Yeah, but my recommendation is if they can stop smoking, that will be a better thing to do.
Absolutely, I agree with that. That's well been proven. Okay.
I will sort of give you an analogy, if I may, about the expression. Now, we all wear clothes, and so we, in this society, when people walk around, and you don't notice people too much when they're wearing clothes. The moment somebody take off their clothes and strip, and all of a sudden you notice them. And that's how, those abnormal cells express themselves when they, sort of strip of some of the sugars on the surface and then exposing the mannose, then that's when the immune system will recognize them and start to take care of them. And so sort of that's how you look at expression.
Okay. I have another question. This was one I know will be right down your alley. Could socket be used for squamous cell cancer inside the mouth? A friend's father had surgery today to remove a part of the jaw and palate, followed by plastic surgery. Could socket help with pain and healing or in any other way?
It'll certainly help with pain and healing. And we have a lot of patients that use it for that.
Yeah.
Now,
the socket is designed for all oral wounds. So any kind of oral wound, it will help. It will help in many different aspects of the healing process. It controls pain as well as hemostatic effect, along with the protective wound, sort of like a wound dressing effect, along with helping to develop a optimal environment for the healing process.
So in the surgery, when they remove the cancer, what you want to do is help promote the healing and turn on all the macrophages to go through and pick out any atypical cells so you don't have a recurrence.
okay. I wanted to, should people who want to find out more about socket or possibly order socket, can they use the same 1-800-755-0044?
Well, the thing is, socket is a professional use product. What you need to do is tell your doctor. We are working on making one that would be, an over-the-counter product. But right now, the one we have requires a professional. So you can ask your doctor to order it, and we'll be glad to talk to him about it.
Should the professional use this same number?
Yes.
Okay. I also wanted to mention that anybody that buys a book that has heard about this book from this webinar should tell the people when they answer the phone here at 1-800-755-0044. So Dr. McAnalley and Dr. Danny's son will know from whence you heard about the book. And so you might want to do that. Let me see. I think that I'm about the end of the questions.
Oh, there are a few more, Doctor. Let me just see if I can get some.
Okay, well, take off what we got.
Okay.
I've gone through everyone I've got.
Folks, I just wanted to mention this. That there were lots of questions about product-specific questions, and as Dr. McAnalley mentioned, he would rather stay away from product-specific questions, so I didn't include those when I sent them to Dr. Marvin. Sorry about that.
Okay. Okay.
I'm sending you some right now. So.
Okay. As soon as I get them.
Okay. And Dr. McAnalley can tell us when you want to stop, because these questions just keep on coming in.
Dr. McAnalley, I have one here. It says, great presentation. I'm curious to know a little more about the injectable acetaminophen. Is this available in the United States?
Yes, it's available for veterinarians. It's a prescription veterinary product. Normally what happens when you come out with a new product in your prescription, anytime you have an injectable, it requires a doctor to make the diagnosis, because now you have, millions of cancer cells, and this works differently. Injectables are not dietary supplements.
Okay. Okay, I'm still looking through this. And one of them wants to know, how soon would I expect Aloe to restore brain cells that were damaged in a stroke? How quick do these work is the question.
Well, that's a good question.
Only God knows, you said before.
Well, the thing is, when you have a stroke, people get most of what they're going to get, the first 6 months. And, good nutrition can keep producing stem cells. And I know people who, take this product have benefits for a long time.
Okay. I tell people that taking these glyconutrients as a supplement or a replacement in what should be in everybody's food that have been taken out by processing.
That's right. But you have nothing to lose, and I would take anything I thought might help.
Absolutely. I've had people that took these glyconutrients for one reason, but ended up having another one solved that I wasn't even aware that they had. And they were rejoicing of how it helped them in one manner that they haven't even mentioned to me.
I hear that all the time.
I bet you do.
I know Danny does too.
Great. I'm just about at the end of these questions, David.
You might want to scroll down.
I am right now.
Okay. I've sent you a few more. Dr. McAnalley, there's a question here. Will you autograph your book for us if we ask?
Yes. Be glad to.
Well, any instructions as to
Just
that
tell
regard?
it to you. I'd like to make it out to you if you want to say, please, my name is Jane Doe. Then I'll say, dear Jane, and write something in there.
Okay.
There's a few quotes I like, like Numbers 24:6, where the Lord planted the aloes and the cedars. It doesn't say planted carrots, onions, or anything else. It singles out those two plants. And then Revelations, 22:2, that the plants are for the healing of the nations. And I really I like those two quotes. But I'll be glad to personalize it.
There are some questions about GMO crops. We hear so much tinkering with GMO crops. Can we test to see if the aloe is genetically modified?
As far as I know, there are not any genetically modified aloes.
Okay. I have another question here.
That's a lot. That costs a lot of money. And I don't know of anyone doing research in that area.
Okay. I have a statement that I don't agree with, but I will put it out here because it came in. It says, do not take soy as it blocks thyroid hormone and is very detrimental to your health in many other ways. Please check out www.wholesoystory.com for the facts.
Yeah, I've read that. I've read that story. And, I know people that look pretty healthy to me and they live on soy.
That's right. Chinese.
Yeah, I'm sitting right next to one right here. And he's brilliant, so if that affected him,
Pass the soy.
I eat a lot of soy, yeah.
So do I.
I think that there are other things that, need to be considered.
My experience, I asked Reg McDaniel, Dr. McAnalley, once, and he said, well, for 4,000 years, the Chinese have been using soy products for cancer effectively. And that was the end of my questions to him.
Yeah, there's so many more well-controlled studies that say it's good for you.
Yes.
Yeah.
He sent me about 84 PowerPoints proving the point.
Well, if you'll do a Google, you'll find there's probably 1 million of them.
Right. I have another question. Go ahead.
You have 1 million of them that say it's good and 1 or 2 that say it's bad. You need to look at those 1 or 2 a little closer.
Right. It says, I may have missed if someone answered the question, how concentrated can these aloe powders be made theoretically? How much more concentrated compared to products in the market can it theoretically be made?
Yeah, you broke up on that. Could you repeat that?
Yes, they want to know how concentrated can these aloe products be made,
Yeah,
200,
200 times and you have a powder. And that's all covered in processing aloe.
Right.
Around chapter 4.
Chapter 4 covers that in the book. Okay. I have another one. Are you taking ACE just mannan or a composition containing other sugars?
I take a composition of other sugars, but I also take a lot of ACE mannan.
Now, when you take ACE mannan, there's a question I've had asked. Is there any other monosaccharides in the product ACE mannan other than mannose?
ACE mannan is, and that's in the book too, it gives the structure and it tells you the other sugars. If you isolate the ACE mannan, you have about 35 sugars of mannose, one sugar, one glucose, and one lactose.
Okay.
Someone is asking if are you taking just ACE mannan or a composition containing other sugars? Is this specifically ACE mannan you take?
You'd already answered that.
Oh, okay.
Isn't that right, Dr.
And,
McAnalley?
and everybody keeps trying to get me to name some products. So, they keep asking the questions different way. I feel like I'm being cross-examined by Perry Mason.
I'm on your side, Dr. McAnalley. I've been screening a lot of the questions over here. But I guess they don't want you to, I'm not sure they're asking you to name the product exactly, but just want to know if you are taking a combination.
Yes, I do.
Okay. There you go, folks.
Yeah. I think the point is that there are a lot of nutrients in the nature that is all important for our body. And today we're talking about, aloe in particular, and that's why we're talking about ACE mannan. And we talk about, the effect of that in our body and why is it so important historically. But that doesn't mean that other nutrients are not important.
You need a balance. In fact, we cover that in our book too.
Right.
Yes.
I think we covered the different classes of nutrients, what you need, the physiology, how they work, how they support structure and function, and in the first chapters, last time we went through, like that's in the physiology section.
Okay.
It all boils down to this, folks. You need to buy this book.
Right.
It's essential if you're using glyconutrient technology. You can order it by 1-800-755-0044 and let the people know you heard about it on this webinar.
Chapter 5, is aloe's effect on physiology, structure, and function. And to show how it works, we had to cover, the vitamins, minerals, lipids, proteins, essential proteins, fats, phytohormones, antioxidants, and carbohydrates, and then how aloe vera fits in with all of that.
Right.
And I think it's interesting. When we first wrote this book, it was probably 2,000 pages long, and nobody's going to read a 2,000 page. But we covered the physiology and all of these nutrients with the references and everything in about 6 pages. So if you want to read if you want to read and understand nutrition and a general concept, I think we did a great job. And that's what I've always looked for. Like I have books. I have, like you're talking about, I have books that talk about, I have 1,000 pages on just proteins and how they work.
Well, I tell you, Dr. McAnalley, this book is written so well, so easy to understand. I would be glad to try the 2,000-page variety just because I know how you and Danny and the rest of your staff worked so hard to make this understandable.
Mm-hmm.
Well,
But,
we spent a year cutting it down to the size it is. And a picture is worth 1,000 words.
Yes.
Sure.
Yes. Yes.
I don't see any more questions, Danny.
I think that covers it for the most part. So I just want to say thank you again. I mean, this has been a very, very well-treated topic. I've learned a great deal. And folks, just like our presenters have said, and you really have to pursue a whole health approach. And one of the fundamentals that you really have to understand, of course, is how to strengthen your immune system. And Manos, Ace Manon, is one of the key, I would say, is one of the linchpins in that whole process of helping to strengthen your immune system. So again, I want to thank Dr. Marvin for being on our webinar and helping to host this. Dr. McAnalley, Dr. Sun, and Eric too. Thank you so much for your presentation. Does any of you want to add anything else before we end the webinar?
I'd like to add, we're standing by to take orders, so now's your chance to call. Last time the phone, we had 3 lines, they were ringing off the wall. And then,
oh my goodness.
We're here now.
Okay, great. So folks, you need to call in, call your friends, ask them to call in, and the number again is 1-800. I'm taking it off. Let me bring that up again one more time.
It's 1-800-755-0044.
755-0044. Thanks, Eric. Okay, great.
I want to say thank you, David, for allowing me to be involved in this. It was a real treat. And thank you, Dr. McAnalley and Danny and the rest of your staff for writing this book. It was wonderful.
Absolutely. Absolutely. Well,
Thank
folks,
you, Dr. Marvin.
Yeah, thank you for inviting us.
I'm sure we'll be hearing a lot more from you in the future, too, as well. Well, folks, this ends the webinar for tonight. Thank you for joining us, and see you on Thursday for an interesting presentation by Dr. Steve and Dr. Harry. And remember, the foundations have to be laid. You need to build upon the foundation. You need to understand everything that has to do with health, not just pieces here and there. So God bless and have a great night.
How do I shut this down?